Again. So quick update on my progress on my gene therapy project, or better said, lack thereof. So I have did a transfection, as I've told you, in 24 wells. These are my plasmids right here, and this is the wells with the control. Would be the positive control, so this would be a plasmid that is known to generate green fluorescence. This one is pcDNA 3.1 GFP. So this is a plasmid that it's widely used in mammalian cell transfection. So let me show you how I've set up my experiment and tell you basically what went wrong. So this is how I have set up my plate. These are the plasmids, each with its own designation number. I used HepG2 cells, which are a type of hepatic cancer cells, and I grew them in 500 microliters of medium with DMEM supplemented with glucose and PBS, right? I've seeded them at about 100,000 cells per well. I've transfected the next day using PI with a ratio of 1 to 3. So that means I used 1 microgram of DNA for every 3 micrograms of PEI. And the next day, so after about 24 hours, I've observed all the wells under the microscope, both optical and fluorescent. The problem is that I did not get any green cells, so none at all. The positive control, which remember was supposed to generate fluorescence and was supposed to transfect, actually did not. I only seen like 2 or 3 green cells for the whole sample. So I have a general idea what went wrong, although not everybody would agree with this. However, I'm gonna test this out in my next experiment, and I'm gonna use a different transfection reagent because, in my opinion, this PEI was not as efficient as expected, because HepG2 cells are, from my knowledge, cells that are pretty difficult to transfect, especially with PEI. So for the next experiment, I'm going to set up the plate in pretty much the same way, and I'm going to be using a reagent from Polyplus transfection, which is called Jet Optimus, and then I'm going to keep you updated with what happened.